Right, Zoe, over to you.
Thank you. So hello, I'm Zoe Morland. I'm a PhD student at the Open University, and I'm here to discuss whether Phobos, one of Mars's moons, could host Martian biomarkers. And if so, why is this important? I'll first introduce Phobos and the motivation for my PhD, and then feed into some modelling work that I've done. So firstly, Phobos is the larger and nearer of Mars's two moons. It's roughly 22 kilometres in diameter. And to put that into context, it's about the size of Manchester's Ring Road. It orbits Mars just beyond the Roche limit in a degrading orbit, meaning that one day Phobos is likely to disintegrate under Mars's gravity and either fully dissipate or coalesce into another moon again. It also orbits Mars subsynchronously, which means that it orbits Mars faster than Mars rotates. In fact, it orbits Mars 3 times every Martian day. A lack of in situ investigation has limited our knowledge of Phobos' surface to reflectance spectra. 2 spectral units have been identified, one redder than the other, shown in the steeper spectrum on the right. Therefore, they were so named the red and blue units. The red-sloped, generally featureless nature of the spectra suggested that Phobos was composed of a D or T-type asteroid or a carbonaceous chondrite-like composition. Under closer inspection, 2 absorption bands were identified, one at 2.8, which is thought to be associated with metal hydroxides, and another, just in the red unit, at 0.65, thought to be indicative of iron-bearing phyllosilicates. This information suggests that Phobos could be composed of a D or T-type asteroid-like material, including metal hydroxides and iron-bearing phyllosilicates. However, the addition of material from external sources, such as Mars, could make Phobos more interesting than its bleak-like surface suggests. If an impact were to occur on Mars, a considerable amount of material could be ejected into the Martian atmosphere. Even though Mars's atmosphere is not as thick or as dense as Earth's atmosphere, material is still likely to experience noticeable ablation. This leaves a considerable amount of material to survive and expand into a large ejector plume spreading towards the orbit of Phobos. By the time it reaches Phobos' orbit, the plume is likely to occupy a significant proportion of Phobos' orbital path. And if Phobos happens to align with the ejector plume, then material could impact the Mars-facing hemisphere. The plume then continues to expand, but comparatively slower than Phobos' orbit, allowing Phobos to sweep up material on its leading edge. Material accelerated to more than Mars's escape velocity of 5 kilometers a second would be lost from the system, perhaps to come to Earth as Martian meteorites. However, material not reaching the escape velocity would begin to fall back to Mars again, where it could potentially impact the non-Mars facing hemisphere of Phobos. Overall, there are multiple opportunities here for ejector from Mars to land on Phobos. Past numerical simulations into this impact-driven transfer of material from Mars to Phobos have yielded the following conclusions. An impact as small as 2.9 kilometers in diameter could eject material which could land on Phobos. However, material less than or equal to 10 centimeters in diameter are likely to be slowed down by Mars's atmosphere, so it may not reach Phobos. The minimum ejection velocity to reach Phobos was found to be around 4 kilometers a second, and owing to the variation in ejection angle impact site on Mars and the locality of Phobos in its orbit, the location of where material could land on Phobos will vary. And it could be as low as 580 meters per second landing on Phobos. And it was suggested that an average of 200 parts per million of Phobos's regolith could be Martian in origin. And that finally, 100-gram sample of Phobos's regolith could contain as much as 50 milligrams of Martian material, of which 0.2 milligrams are likely to have been deposited on Phobos within the last 10 million years. Together, all of these conclusions suggest that Phobos could be a concentrated record keeper of Mars's past. This is of interest because of planetary protection, which is an internationally agreed practice defined by Article 9 of the UN Outer Space Treaty, which states that parties to the treaty shall pursue studies of outer space, including the moon and other celestial bodies, and conduct exploration of them so as to avoid harmful contamination and also adverse changes in the environment of Earth resulting from the introduction of extraterrestrial matter, and where necessary shall adopt appropriate measures for this purpose. The Committee on Space Research, otherwise known as COSPAR, develops policies to adhere to this treaty by mitigating forward and backward contamination as a result of space exploration, to ensure that scientific investigations of possible extraterrestrial life forms, precursors and remnants are not jeopardized, and that Earth must be protected from the potential hazard posed by extraterrestrial matter carried back by spacecraft. Each mission is characterized depending on its risk associated with the target body and the mission type. So category 1 is any mission to a target with no astrobiological relevance, such as Mercury or the Sun. Category 2 is any mission to a target that has significant interest relative to chemical evolution or the origin of life, however, that there is only a remote chance that contamination carried by a spacecraft could compromise future investigations. This includes targets like the moon and gas giants. Then there's category 2 star, which is an extension of number 2, to targets where there is a remote potential for contamination of liquid water environments, such as the icy satellites like Ganymede. Category 3 and 4 are flyby and orbiter and lander and probe missions, respectively, to targets of significant interest and also significant chance that contamination could compromise future investigations. This includes Mars and the moon's Europa and Enceladus. Specifically for Mars, there are 3 subdivisions of category 4. 4A being landers not carrying any instruments for investigation of extant life, and 4B for landers that are designed to investigate extant life, and then 4C, which are missions to special regions, which have been classified as sites where terrestrial organisms are likely to replicate, and any region with a high potential for the existence of extant Martian life. For each increase in planetary protection categorization, requirements for outgoing spacecraft sterilization are more and more stringent, with controlled bioburden levels. Finally, category 5 are for Earth sample return missions, the first being unrestricted, which is where the target is deemed to have no indigenous life forms on the surface, such as the moon and asteroids Itokawa, Bennu and Ryugu. In this situation, planetary protection requirements are only required on the outbound phase, typically in category 1 or 2. Secondly, there is restricted Earth sample return, where samples are likely to be of significant interest to chemical evolution and the origin of life, such as Mars and the moon's Europa and Enceladus. In this situation, outgoing spacecraft have high levels of planetary protection measures, and the return samples and the spacecraft are required to be sterilized and or quarantined, among many other expensive protective measures to ensure that forward and backward contamination is mitigated. This is all of interest for Phobos because if an impact were to occur on one of the special regions of Mars, then Martian life in any form, from living organisms down to the breakdown products such as biomarkers, could be incorporated into the ejector and delivered to Phobos. Therefore, a portion of the 250 parts per million of Phobos's regolith, which is Martian, could also include astrobiologically relevant material. The possibility of this process raised questions about whether or not Phobos should be treated in the same planetary protection restrictions as Mars, which became especially relevant with increasing interest of Phobos sample return. Should this be unrestricted like a target body which is devoid of life or contamination issues, or should it be restricted, considering that Martian material and therefore possibly Martian life signatures could be present on the surface of Phobos? This motivated two study groups to investigate whether enough living microbes could survive sterilization during impact-driven transfer from Mars to Phobos, to result in that more than 1 in a million chance of collecting Martian microbes in a sample taken from Phobos. And if this is exceeded, then this would require a higher level of planetary protection classification. ESA ran lab impact simulations using a light gas gun, as well as isolated heating experiments, to investigate whether the heating alone was the dominant organism sterilization process during impacts. They also considered sterilization from radiation exposure on the surface of Phobos after deposition. These simulations fed into two models of sterilization, one of impacts dominated by heating and the other from radiation exposure. JAXA expanded on the Stirling models by taking into account sterilization through impact ejection, aerodynamic heating as ejector ascends through the atmosphere, and also the post-deposition meteoroid environment. Both studies came to the same conclusion, that the processes involved with impact-driven transfer of microbes from Mars to Phobos and the subsequent existence within the Phobos regolith are not favourable for the chances of sampling Martian microbes to be higher than 1 in a million. Therefore, future sample return missions, such as JAXA's MMX, which is the Martian Moon's exploration mission, aiming to launch in 2024 to collect and return samples from Phobos, can remain as unrestricted sample return under planetary protection. Now microbes have been reasonably ruled out, the question arises of whether Phobos could still be astrobiologically relevant. Both of these studies used terrestrial analogue organisms from the Atacama and Antarctica to inform the biological loading of Martian material being ejected to Phobos. However, this neglects non-living yet still astrobiologically relevant materials, such as biomarkers, which could be included within return samples and possibly even in usable concentrations. This was the foundation of my PhD project, which is to investigate whether just the biomarkers could survive different stages of impact-driven transport from Mars to Phobos. From the ejection from Mars, ascension through the atmosphere, and then deposition onto Phobos. This takes a step back on the organism complexity ladder and instead considers compounds that represent the building blocks of future life or the resistant breakdown products of past life, and therefore components that could possibly have been preserved for billions of years on Mars or Phobos. The initial intention was to simulate the stages of transfer almost entirely practically by running a series of impact and heating experiments to simulate biomarker ejection from Mars, aerodynamic heating through the atmosphere, and final deposition onto the surface of Phobos. And the impact experiments were going to use the all-axis two-stage light gas gun at the Open University. However, the world had different ideas, and as I was about to begin my lab work, COVID-19 appeared and the associated national lockdown. So this lab work has been put on hold. So instead, I looked towards modelling, bringing forward a collaboration with Sam Halim, a PhD student at Birkbeck University of London, who uses ISAIL shock physics code to investigate biomarker transfer from Earth to the Moon. Impact modelling in comparison to practical experiments can record information from within the system which is practically inaccessible in practical experiments, such as the conditions within the projectile during impact. Therefore, we devised a study to look at just the impact of Martian material landing on Phobos. Biomarkers themselves cannot be simulated in ISAIL. Therefore, the study aimed to investigate whether the conditions within Martian ejector are favourable or not favourable for biomarker survival. This involved estimating the pressure and temperature conditions within Martian-like ejector particles impacting a Phobos-like surface. Furthermore, this modelling addresses the shortcomings of past models mentioned earlier, such as that Martian ejecta has been simulated as point-like, strengthless, and sometimes massless particles with a defined dispersion rate. Instead, this model looks at an individual particle impacting a surface. The model's resolution is represented by a grid of individual cells. And for these simulations, it is 50 cells per projectile radius. Then the model includes tracers, which are massless particles that follow the material as it flows and records information, such as the pressure and temperature throughout the simulation. And these are placed throughout the projectile, one for every cell. And these measure the conditions spatially throughout the projectile throughout the simulation. In these simulations, there were 7,848 tracers over the whole projectile. However, in reality, just half of the projectile and the target is calculated, referred to as the half space, because it is assumed that the other half mirrors the first half. For simplicity and ease of visualization, from now on, I will display the whole projectile and the target, bearing in mind that these actually represent one half replicated to make a full projectile and target. In order to run the model, several starting parameters needed to be defined. Some are reasonably simple. For example, it is assumed that the Martian ejecta projectiles are spherical, which is a reasonable assumption because irregular shaped projectiles are likely to spin, meaning that they can be reasonably represented by spheres. It is also assumed that they impact vertically, which on this scale and considering other assumptions is perfectly valid. And furthermore, that they are composed of one of two materials representing Mars: basalt, the global composition of Mars, and therefore the most likely material to be ejected, or a clay-bearing material such as serpentine, which is a more likely material to preserve biomarkers, and has already been observed to preserve organic material on Mars. However, this is far less likely to be ejected from Mars. And for the Phobos target, it is assumed that the surface temperature is 200 Kelvin, representing the seasonally average equilibrium temperature. And it is composed of a surface regolith layer and a more cohesive interior. Other parameters are, however, more challenging to define because they are largely uncertain. These include the projectile size, impact velocity, and serpentine projectile density. And for the Phobos target, its composition, strength, and regolith depth. Some of the above unknown parameters have estimated ranges, but no reasoning to choose one value over another. Furthermore, restrictions within ISAIL present further limitations. Together, these culminate in a large matrix of different possible ways to represent Martian ejecta landing on Phobos. And if I wanted to simulate all of these, I would require a whole other PhD to do so, and perhaps even longer. So this led to a series of tests to narrow down the possibilities by investigating the influence varying each parameter has on the resultant pressure and temperature conditions within the projectile during impact. If the influence is minimal, then the specific value I choose is insignificant, meaning that one representative value can be chosen for all of them. However, if the influence of varying the parameter is large, then the choice of value will strongly affect the resultant conditions within the projectile, and therefore my interpretation of biomarker survival. Taking Phobos's composition as an example uncertainty, our best approximation is a D-type asteroid, therefore carbonaceous chondrite-like composition, as we saw earlier. An exact match for Phobos is not available within ISAIL because this requires an equation of state to be defined using shock experiment data. However, the equation of states previously used to represent carbonaceous chondrite-like composition include basalt and serpentine. Another relevant option is dry tuff, a basaltic ash, because it is common for airless bodies like Phobos to have an anhydrous regolith. No one of these materials better represents Phobos than another. So the different combinations of these three materials are used in the regolith and the interior in different combinations to test in the simulation on the right, to test how the different variables of the different materials correspond to differences in pressures and temperatures. Having run the simulations for the pressure and temperature results, these can be superimposed onto the original shape of the projectile in contour maps to show how conditions vary spatially. Again, to reiterate, these maps represent only one half of the projectile mirrored and spliced together to make a whole projectile. Specifically, these maps display the peak conditions, so the highest pressure and temperatures reached at specific points within the projectile during the entire simulation. Firstly, here is peak pressure, where yellow expresses high pressures and purple-black lower pressures. The bottom is the leading edge of the projectile, which is the first point to come in contact with the target, and therefore undergoes the most extensive compression. And as expected, this region experiences the highest pressure, indicated by the light yellow region. The scenario that sticks out is number 3, which is where the target is tough overlain with tough, and this displays a much smaller region of high pressure at the front of the projectile, and lower overall peak pressures across the whole projectile. This is more obvious on a graph showing the volume of projectile for each material scenario reaching specific pressures. It shows that dry tough overlaying dry tough, in pink, has consistently lower volume of projectile reaching each pressure. Other material combinations here are hard to distinguish from one another. Now looking at peak temperature, number 3, dry tough again, sticks out as having the largest volume of low temperature material in purple. A greater difference this time can be seen between the other material combinations, showing that number 4, basalt serpentine, and number 6, basalt both have large volumes of high temperature material in yellow. Again, comparing it to the graph of volume to peak temperature separates the scenarios more, showing that the scenario where the target is dry tough is consistently the lowest temperature over the whole of the volume. Others containing dry tough also have much lower volumes at higher temperatures. Basalt serpentine, however, shows up clearer here as in light green as the scenario where the highest volume reaches the highest temperatures. This test demonstrates how different target material combinations strongly influence the pressure and temperature conditions experienced within the projectile. Owing to this work being focused on the survivability of biomarkers, it makes sense to consider both the best and the worst case scenario for biomarker survival. From shock heating literature, I found that biomarkers like amino acids and fatty acids are likely to survive up to around 500 to 600 degrees C. I've highlighted this threshold range in Kelvin in the shaded region. To the left of the region, biomarkers are more likely to remain intact, and to the right, decomposition and racemization is more likely to occur. This clearly distinguishes that the different material combinations alone generate more and less favourable conditions for biomarker survival, i.e. there are different volumes of the projectile reaching the different temperatures and reaching the threshold temperature. So tough is the best case scenario because the smallest volume of the projectile reaches the threshold temperature, whereas basalt serpentine is the worst case scenario because the largest volume of the projectile reaches the threshold range. A conclusion can be drawn here that even when all of the other parameters are constant, all scenarios of target material combinations result in at least 20% of the projectile volume remaining below the temperature limit at which deformation and racemization of biomarkers are likely to occur. This is very encouraging because it suggests that in every situation there are areas where biomarkers could survive. So after this test, I can take forward the 2 outermost target material combinations as the 2 variables which are the best and the worst case scenarios, as these bound the extreme conditions which are controlled by the target material. I ran tests like this for different estimated parameter ranges to identify the best and worst case scenarios for biomarker survival for each of the uncertain parameters, yielding the following values. Firstly, the best and worst case target scenarios vary in material composition for each layer, regolith depth, which is deeper for the best case scenario than the worst case scenario, and then density, which is controlled by the material and the constant layer macroporosity. And secondly, for the projectiles, there is just one basalt projectile scenario because this didn't have any parameter values ranges to test. But the serpentine has a variation in macroporosity and therefore density, so this has a best and worst case scenario. In summary, the main differences between the best and worst case scenarios for the target are material, regolith depth and density, and for the projectile are material, density and macroporosity. These variables feed into the main set of simulations to investigate how the best and worst case target and projectile scenarios combined with the final continuous uncertainty, which is impact velocity, result in different temperature and pressure conditions within the projectile during impact. The actual vector impact velocity has many different factors, including initial impact location on Mars, the ejection angle and its velocity, the location of Phobos within its orbit, the intersection point with the ejector plume, and Phobos's orbital velocity. However, for this study, I need to hone all of these down and define an impact velocity range to test. Modelling has suggested that material must be ejected at 4.2 kilometers a second to reach Phobos's orbit. But owing to the trajectory and deceleration from the atmosphere, material could impact Phobos with a minimum velocity of 0.58 kilometers a second, as seen earlier. The maximum velocity for material to land on Phobos is limited by the original impact velocity on Mars, which could be tens of kilometers a second for cometary impacts. However, studies have shown that material ejected at more than 10 kilometers a second is likely to be almost completely molten, which is not favourable for biomarker survival, which is the focus of this study, so this can be placed as an upper limit. Finally, the inbound ejector, which was unable to escape Mars, then impacts the non-Mars facing hemisphere, will be travelling at lower than Mars's escape velocity of 5 kilometers a second. Considering all these values, as well as the ejection angle, impact plume intersection point, and Phobos's orbital velocity contribution, an average impact velocity range has been estimated at between 2 and 4.5 kilometers a second. So in this final simulation series, I chose an impact velocity range of 0.5 to 8.5 kilometers a second at 2 kilometers a second intervals. It's not necessary at this stage to simulate impact velocities higher than 8.5 kilometers a second, because above around 5, the likelihood of occurrence drops off significantly. And this also tends towards conditions which are more unsuitable for biomarker survival. So the range chosen here covers the possible impact range for Phobos and also falls within the realistic boundaries of biomarker survival. So this resulted in a total of 30 simulations where the 5 different impact velocities were simulated for the basalt and serpentine projectile and for the best and worst case target and projectile parameters. It should be noted that for all of these tests, the projectile size remains at 1 meter. It was the intention to test multiple projectile sizes down to around 10 centimeters. However, this became computationally expensive and would have required more time. This could be addressed in the future though. I have just received the results from these simulations, so here are my initial thoughts about the average peak pressures and temperatures reached within the projectile over the duration of each of these simulations. Firstly, just for the basalt projectile simulations, what is immediately clear is that, as expected, with increasing impact velocity from light green to dark blue, the pressures and temperatures within the projectile increase, which is as a result of the additional energy supplied to the system by the increase in velocity. This makes sense, and this was expected. Furthermore, what can initially be observed is the difference between the 2 simulations at the same impact velocity, so the difference between the circles and the triangles of each individual colour. which shows that the best and the worst case target parameters result in different temperatures and pressures within the projectile. Again, this was expected. Then for the serpentine projectile simulations, it again can be observed that across the board there is an increase in temperature and pressure within the projectile with increasing impact velocity. However, the additional observation here is that the star and horizontal line data points representing the scenarios with the worst projectile parameters sit on a different trend to the cross and X data points representing the scenarios with the best projectile parameters. The likely explanation for this is the additional macroporosity present in the worst projectile parameter scenarios, which introduces the requirement for pore collapse during impact, which in turn slows down the shock waves, resulting in lower average shock pressures, and generates isolated spikes in temperature, where pore spaces are closing. This results in the higher average temperatures. This phenomena is so significant, in fact, that a scenario with a porous projectile, like the one highlighted in the pink circle, generates temperatures 200 Kelvin higher than a scenario with zero porosity, but impacting 2 kilometers faster, highlighted in the yellow circle. Now representing all of the scenarios on one graph reiterates the difference between the porous and non-porous projectiles, irrespective of projectile material, showing that a porous serpentine projectile generates higher temperatures than both the non-porous basalt and serpentine projectiles. The last initial observation I can make is by observing the biomarker temperature survivability threshold from earlier, which shows that generally impact velocities above 2.5 kilometers per second generate conditions unfavorable for biomarker survival. However, these values here on the graph represent the mean peak temperatures and pressures over the whole of the projectile. Therefore, even at the highest impact velocities, there may be regions within the projectile that could experience temperature conditions lower than this threshold range. This is going to be my task to look for these later, so I'll leave this on a cliffhanger for later. So to summarise, I've embarked on a modelling study to investigate how different parameters within the realistic boundaries of Martian-like ejector landing on a Phobos-like target control whether the conditions within the ejector are favourable or not favourable for biomarker survival. From my preliminary results, I can make the initial conclusion that there are several scenarios under which biomarkers could survive, suggesting that this process of biomarkers transferring from Mars to Phobos is possible. After processing all of these results, I will be able to go on and estimate which specific Martian relevant biomarkers could survive the journey from Mars to Phobos, and therefore I could imply which biomarkers could be collected by future sample return missions, such as JAXA's MMX mission. I shall leave it there and welcome any questions.